thanks for still being here, real troopers. And my group developed large-scale spiking network models of the cerebral cortex that we hope others will make them publicly available, and then to build upon and ask all kinds of questions too. And these models, they relate the structure of cortex to their dynamics. We are very interested also in brain function, of course. We want to understand how the brain works and processes information. But we focus in the first instance on this relationship between structure and dynamics for a reason that I tried to illustrate with this analogy. So consider this creature, this insect called a water strider. If you want to understand how the water strider manages to walk on water, then you have to know about a property of water, namely surface tension. If you didn't know about the physical property of surface tension, then you would come up with the wrong explanation for how the water strider manages to do this. For instance, just thinking, oh, it's particularly light or something. And by the same token, if you kind of modeled the system in the wrong parameter regime, say in a soapy regime, then you would predict that the water strider would drown. And by the same token, if we want to understand function, which by analogy is here the water strider that kind of rides on top of the physical substrate of the cerebral cortex, then we also need to model cerebral cortex first with the proper mechanisms and in the proper parameter regime. And our models, they're based on a collation of extensive experimental data, anatomical physiological data. And they're multi-scale, going from the single neuron spiking activity to the interaction between multiple cortical areas. And maybe you don't need to be convinced that this is a useful thing to do. But in any case, in computational neuroscience, there's sort of a zoo of models that tend to focus on individual phenomena, but we're not coming together as a field to develop more integrative models that account for an increasing set of phenomena. And that are validated with experimental data at multiple scales, and that can account for the origins of these signals at different scales, like the local field potential, the EEG, the fMRI signal, and at the same time use those signals to constrain these models more and more. So we would like to get there. And our models, they use the full density of neurons and synapses in each local circuit that we model, which is enabled by the simulation technology and the access to high-performance computers that we have. And a reason for using the full density of neurons and synapses that we investigated via mean field theory some years ago is that when you downscale the numbers of neurons and synapses, then even if you try to make up for it by adjusting parameters, for instance, increasing the strength of synapses when you reduce their number, you necessarily change the dynamical predictions of your model. For instance, the correlation functions. Correlation function just being the correlation between neural activities of pairs of neurons as a function of the time lag. These correlation functions, they change their shape when you downscale too much. And when you reduce the number of neurons in your simulation, then you tend to increase the correlations between the neurons. And you can understand this intuitively by considering that if you have a certain fluctuation in activity in a smaller network, it has a relatively larger influence on the rest of the network compared to in a larger network. Okay, so as far as possible, we try to model the natural density of neurons and synapses, which leads to really huge numbers of neurons and synapses, of course, when you're simulating mammalian brains. A building block for our models is this cortical microcircuit model that was developed some years ago by Potjans and Diesmann. And it's a model of early sensory cortex, kind of combining data from visual and somatosensory cortices. It covers a square millimeter microcircuit, and it has a full density of neurons and synapses, leading to about 80,000 neurons. And we use point neurons, leak, integrate, and fire models. And they are connected via about 300 million internal synapses. In each of the cortical layers, there's an excitatory and an inhibitory population of neurons. They're here modeled with identical intrinsic properties to focus more on the influence of the connectivity structure on the dynamics. But what is sort of very biologically based in this model is that is the cell type specific connectivity that is based on really extensive anatomical and physiological data. The anatomical data is light microscopy data, where the connection probabilities are based on something that we call Peter's rule, or some version thereof, that basically assumes that the connection probability is proportional to the overlap in space between the axonal and dendritic trees. And the physiological data is their paired recording. So from the literature, a huge number of studies, like 50 different studies. And the model just reproduces the asynchronous irregular spiking activity of the cerebral cortex with low rates that are population specific and layer specific. So for instance, in this raster plot on the right, you see spike times of excitatory neurons in blue and of inhibitory neurons in red. And the inhibitory populations have slightly higher rates than the excitatory counterparts in the same layer, despite being modeled with identical intrinsic properties. And for instance, layer 2-3 has low rate and layer 5 has relatively high spike rate. In this model, the connectivity had to be a bit tweaked compared to the experimental data. In particular, the specificity for inhibitory targets had to be manually increased to obtain this robust low-rate asynchronous irregular activity. So it kind of provided a prediction that we weren't recently able to verify using electron microscopy data, which I will show in the next slide. But first, this model is now, it's in wide use in computational neuroscience, and it has been made available on different platforms, different kinds of software and hardware, and we ourselves were also involved in porting it to the neuromorphic hardware Spinnaker. So now this sort of an intermezzo, because we don't use the electron microscopy microcircuit model for the rest of our work, but it's a recent study by two former PhD students that I worked with on the microns data set on mouse visual cortex. And here you can, lots of neurons were actually really precisely reconstructed, and then you can actually see where the synapses are, and it provides more of a ground truth compared to light microscopy data that doesn't image the individual synapses. And here is plotted confusingly in the opposite color scheme compared to the previous slide. So here, blue refers to more inhibition and red to excitation. It's more the water tap notation where like red is warm and blue is cold, let's say. And here in the light microscopy data set, if you don't do this manual adjustment, you have a preference for excitatory targets. And in the electron microscopy data set, you have preference for inhibitory targets. So if we want to use light microscopy data, we have to increase this specificity for inhibitory targets, and this kind of confirms this prediction of the Potjans-Diesmann model. And you can see that naturally in the electron microscopy data, when you simulate a model based on that, then you don't get these. You get very asynchronous activity, whereas when you use the light microscopy data without further adjustment, then you get vertical stripes in this raster plot corresponding to synchronous activation of different neurons. And also this, you can see it when you adjust the external drive to the excitatory neurons and to the inhibitory neurons. Then in the light microscopy based model, you have some region with very high firing rates and high synchrony, whereas in the electron microscopy based model, there's robust low rate asynchronous irregular spiking activity. Well, going back to our modeling, this microcircuit model of Potjans and Diesmann, it's nice, but it still misses a number of things that are seen in cortex. For instance, low frequencies and spiking activity with that you can see in parallel recordings of single neurons, or in the spectrum of the spiking activity. That so you have relatively high power at low frequencies. At least here, these are both in lightly anesthetized monkeys. And of course, because it's a single area model, it doesn't reproduce interactions between areas as seen in fMRI resting state networks, or propagation between areas. For instance, seen in an EEG-based study here, where there was increased parietal to occipital propagation of activity in visual imagery compared to visual perception. So in the perception condition, the subjects were just watching videos, and in the imagery condition, they were imagining or trying to recall those images, or they were imagining like riding a bike without pedaling. Yeah, so you can think that in the perception condition, you have more sort of bottom-up visual activation, whereas in the imagery condition, the activity is coming more from your brain itself. And then in this sort of spontaneous imagery condition, there was increased flow from the parietal to the occipital regions. When creating multi-area model, we focused on macaque visual cortex for a few reasons. So very rich anatomical and physiological data sets are available for this system. Macaque is relatively close to human, which we ultimately may be most interested in anyway. In one hemisphere, all these 32 areas, vision-related areas of the macaque cortex, they contain about 800 million neurons, which is still too many to simulate routinely, even with simulation technology of NEST and the supercomputers. So we represent each area by a square millimeter microcircuit, preserving the relative in degrees of the Potjans-Diesmann model. So each pair of populations has a certain connection probability, and that goes along with a certain number of incoming synapses per target neuron coming from that particular source population. And in this model, we preserved that because we kind of thought, so the in degrees are the most determinant of the spike rates. So if we want to have similar spike rates, reasonable spike rates, that might be an easy way to go. Later in a human cortex model, we made a different choice, but I just mention it here. So modeling each of these 32 areas by square millimeter microcircuit with a full density of neurons and synapses leads to a model with about 4 million neurons connected via 24 billion synapses. And we simulated using NEST on the Jülich supercomputers. As some of you may know, there's an organizing principle of vision-related cortical areas in the macaque monkey. They are organized hierarchically when you consider pairwise laminar connection patterns. So going from a lower area in the hierarchy, like primary visual cortex, to a higher area in the hierarchy, the connections mostly come from supragranular layers, from the upper layers, and terminate in layer 4. Whereas if you go down the hierarchy, then the connections mostly come from the lower layers and terminate outside layer 4. When you're at the same level of the hierarchy, then the connections come about equally from the upper and lower layers, and they terminate in all the layers, what was here described as in a columnar pattern. Then you can try to order the cortical areas in a way that is as consistent as possible with all these pairwise orderings. So you cannot attain full consistency, but you do get some commonalities between orderings that violate the same number of pairwise constraints. And primary visual cortex is always at the bottom of the hierarchy, and then secondary visual cortex, and as you go up towards frontal, prefrontal regions, and here also parahippocampal areas via the ventral and dorsal visual processing streams. But Klaus Hilgetag noted that this is only one of thousands of possible orderings that are equally consistent with the set of pairwise laminar patterns. So an alternative way of defining these hierarchical relationships is using the architectural types, so the cytoarchitecture of the areas. And well, I did not know about Katerina's work yet when we when we did this, but we use cytoarchitecture. Hope you're not angry. So neuron density, it increases with architectural type, and then and it decreases as you go up the visual hierarchy. So primary visual cortex, it has a high neuron density, and it has a relatively thick layer 4, and also very distinct layers. And as you go up the hierarchy, then neuron density decreases, the thickness of layer 4 decreases, and the distinctiveness of the lamination decreases. And at the same time, the volume density of synapses is relatively preserved. There are at least some indications of this. It hasn't been measured all over, but so that in areas with high neuron density, there are relatively few synapses per neuron, and in areas with low neuron density, there are relatively many synapses per neuron. You can think of this also as that the neuron sizes are increasing as you go up the hierarchy, and they take up more space, so their density is lower, but they send and receive more synapses per neuron. So we make this assumption of a constant volume density of synapses. These architectural types, they were determined by Helen Barbas for 14 out of these 32 areas, and we estimate the others just based on the neuron densities. And we also, to determine the sizes of the neural populations, we also need to know how thick the cortex is in each area and how thick the layers are in those areas. And we use some very simple statistical regularities to predict those. And here, looking at the relative layer thicknesses of each area, I looked at lots of cortical micrographs from the literature, so microscopic images, and just kind of measured with a ruler like how thick these layers are, and it's consistent with this idea of the hierarchy. So from right to left, we're going up the hierarchy. The neuron density is decreasing, and the relative thickness of layer 4 is also decreasing, and all the other layers are uniformly becoming thicker. A next ingredient after the population sizes that we need is the connectivity between the areas, and for that we use axonal tracing data that are collected, for instance, in the COCOMAC database. It stands for collation of connectivity data for the macaque brain. And then there are quantitative tracing data from the lab of Henry Kennedy. These are characterized by so-called fractions of labeled neurons from retrograde tracing experiments. So in retrograde tracing, a dye that is injected is taken up by the axons and is transported back to the sending neurons. And then they actually counted all these sending cell bodies. So that gives a very rich data set, but it's still not, it wasn't done at least for all the areas. So we still need some statistical regularities. And we just use this more or less exponential decay of connection density with the distance between the areas to fill in the connection densities where we know there is a connection from COCOMAC. And then when we do this, we arrive at this area level connectivity map. Yeah, that is so stripy. It is also related to mapping between different parcellations, because then for areas where the data is available, they overlap partially with several areas in the other parcellation, and that's why it ends up looking like that. Roughly connected according to this analysis, but more importantly, the connection densities span roughly 6 orders of magnitude. So in the past, quite often, maybe still, binarized connectivity matrices are studied, but it doesn't make a lot of sense when you consider this huge variability in connection density. So now we focused on the area level, but we have the layers, and we also need to determine the laminar patterns of the connectivity. And these tracing data from Henry Kennedy, they're also partly layer specific, and they counted fractions of supragranular labeled neurons. And if you remember, these the laminar connection patterns were different up and down the hierarchy. So fractions of supragranular labeled neurons also vary systematically according to the hierarchical relations between the pairs of areas. And here we just take as a proxy of hierarchy here again these neuron densities, and then do statistical fit so that we can use the neuron densities to predict the laminar patterns. And then on the target side, so for the target area, we know, or we have some prediction of where the synapses are located, in which layer, but that doesn't say yet onto which neurons they are established. The neurons might be in a different layer, particularly in a lower layer, because they extend their apical dendrites up in cortex. And here we again use this statistical mapping of synapses to target cells based on the extent of their dendritic trees in each layer. We use for that CAT-V1 morphologies, because there's a good data set available for that. It's not available for all these monkey areas, unfortunately, but that would be better, of course, if we had specific data for that. After this, we still need to distinguish between the excitatory and inhibitory cells. And once we've done that, we have arrived at a connectivity matrix for our model at the population level. Below the level of the populations, we we connect randomly, so with a given connection probability. There are a few steps before this, actually, that I don't have time to go into, but the model, when you just turn it on, it doesn't immediately give nice activity. It's a complex model developed in such a bottom-up way. It just, you turn it on and it gives nonsense. And we had to use a mean field-based method to slightly adjust the connectivity to enable the external drive to be adjusted and give reasonable spike rates everywhere. But once we've done that, we have sort of base state that actually looks in the pattern of activity still very similar to the isolated microcircuit model. But in the experiment, we had these sort of maybe population bursts of activity of different lengths. And we thought we can introduce these low frequencies by increasing the strength of the synapses between the areas. Those synaptic strengths are anyway not known from experiments, so we're kind of free to play around with them. And when we increase them by factor 1.9 here, we get this mix of slow and fast fluctuations that are also seen in the experiment. When you go even above and have stronger inter-area coupling, then you end up in a high activity state that is not realistic. But just below this transition to this high activity state, we have this dynamical slowing of the activity. And the corresponding spiking activity looks like this for 3 out of the 32 areas, just as an example. And it looks perhaps overly synchronized. But normally, there are never this many neurons recorded in parallel in actual experiments. So when you plot it, it looks less synchronous when you plot the spikes of fewer neurons. To know exactly if this is realistic or not, we compared with the data from this experiment that was published previously. These are just 2 different epochs of the same resting state activity of primary visual cortex of a monkey, where they identified 140 single units. And the overall spectrum of the spiking activity from the experiment is given by that yellow curve here. And from our simulation, it's given by the black curve when determined from 140 random neurons from our simulation. And yeah, you see the same. So the power is approximately the same and also has approximately the same shape. So we're not overestimating the synchrony in our simulation, actually. Otherwise, we would have much higher power. And we also looked at the distribution of spike rates across neurons. So for each neuron, you just count how many spikes it fired in a certain interval, and then you create a distribution across neurons. And at this point, just below this transition to the high activity state, we also reproduce really nicely this distribution of spike rates. If you had weaker inter-area synapses, then it would look like the red curve. And if you had stronger inter-area synapses, it would look like the blue curve. But yeah, just below this instability, it reproduces the experimental data. And this is without actual tweaking to these data at all. So we only required that there was a mix of slow and fast fluctuations. And we didn't fit to these data, but it's a positive validation. We also looked at the propagation of activity across the areas, which we termed temporal hierarchy. We just looked at the pairwise correlation functions for pairs of areas and looked where there is a peak, if it's at a positive lag or at a negative lag, and then ordered the areas in a way that's as consistent as possible with all these pairwise orderings, just by analogy with the anatomical hierarchy. Does this work? Don't know where to click or has it stopped being clickable because I turned it into a pointer?
Change it back to
I see. I don't know if yeah. Okay, well, it's not super informative, but it's the moving part. So, and the activity propagates mainly from the parietal regions to the temporal and back to occipital, and then followed by trough activity in the frontal regions. So in any case, this ordering of the parietal before the occipital regions is consistent with this EEG experiment that I mentioned in the beginning, where there was visual imagery, because yeah, we're modeling the resting state and not some visually driven state. Finally, we looked at functional connectivity as measured with fMRI. On the right is experimental data averaged over 6 monkeys, and the areas are ordered according to the sort of cluster structure that is often observed. And when we have this synaptic strength between the areas where the microscopic spiking activity of V1 was well reproduced, then we also get a pattern of correlations that's very similar to the experimental data. So that's nice. It kind of occurs at the same value of inter-area synaptic strength, and it predicts that the inter-area synapses are stronger than the local ones. But I must say here we had a really basic and actually wrong proxy for the bold signal. We didn't have a balloon Windkessel model or like low pass filtering that should be applied, because our model has the fast fluctuations, but not the slow fluctuations that mainly shape fMRI functional connectivity. So that's something that we should further explore. We made the model code publicly available on GitHub, and now also you can go to eBrains and try out a downscaled version of this model. We're not in favor of downscaling, of course, but not everyone has access to a high-performance computer. And then you can see, at least you can play around with it, and you can see like the connectivity matrix, and then see if you're interested in it, and to try to get access to some cluster or supercomputer to run the full model. So this was all nice, but there's still some things wrong with our multi-area model, besides this bad proxy for the bold signal, which and I will go into the issues, but some of the we still had highly synchronized activity in quite a few populations, and some populations in areas the spike rates were vanishing, and others they were too high. And this work presented a way in which this can be combated. So in cortex, there might be a clustering of excitatory and inhibitory cells together. It's only the excitatory clustering that has been directly measured, but then there's also evidence that there's increased synaptic strengths between pairs of inhibitory and excitatory neurons that are connected. So there's a sort of this motif that of reciprocal connections between E and I neurons that would be consistent with E and I neurons clustering together. And this model is a model of macaque motor cortex developed by Wahid Rostami at the University of Cologne. And it reproduces variability dynamics and reaction times in a center outreach task. I will not go into that, but just the dynamical effects of the joint clustering of excitatory and inhibitory cells makes that when a cluster is selected by giving an extra input, that the firing rate remains moderate, and the spiking activity remains irregular, and so there's a tighter balance of excitation and inhibition when you have this type of clustering, compared here with networks where only the excitatory neurons were clustered together. And we use this insight to further improve our macaque multi-area model. Particularly, Jari Prono did this. So we divided each of the microcircuits into n clusters, up to 50. It's just illustrated here for 2 clusters, where the neurons in the same cluster had increased synaptic strength between them, and between the clusters they had decreased synaptic strength between them. And then the first cluster of area A is preferentially connected also to the first cluster of area B, and so forth. Then this really helped to get rid of this excessive synchrony in some areas, and this silence of some populations, and excessive spike rates of some populations. Here shown in the spike rate distributions for the original model, and here for a model with 50 clusters. And also the propagation of activity when you stimulate area V1. Here in the original model, it didn't even make a bump in activity beyond area V2. But in this clustered model, we can have robust propagation of activity across the areas. And you see in some areas an alternating activation of the different clusters, which can be maybe characterized as winnerless competition. But one cluster becomes active, and then another cluster becomes active and is the winner, let's say, and they alternate. So these stimuli can now, or stimulus-related activity can now propagate across the entire network in the feed-forward direction, in the feedback direction, and the more clusters we used, the more pronounced this became. Another aspect that's reproduced by this clustered model is a decrease in Fano factor upon stimulation, upon visual stimulation. So Fano factor being a measure of trial-to-trial variability. And this was described by Mark Churchland and co-workers some years ago. And so for these 4 different areas, they had data before visual stimulation, and then after visual stimulation, the Fano factor is reduced. So the trial-to-trial variability is reduced. These 2 colors for MT, they just represent 2 different data sets. And in our original multi-area model, this was this phenomenon didn't occur. It was way off. And in the clustered model, it's nicely reproduced. So you can think of, if you have winnerless competition, then you can have high variability when you're not stimulating a particular cluster, but when you're stimulating a particular cluster, and it's the same cluster across the different trials, then the variability across trials is reduced due to this stimulation. Finally, we developed more recently a human cortical model of all the areas in one hemisphere of human cortex, according to coarse parcellation. So the number of areas is not that much higher, just 34. And again, we represent each area by the square millimeter microcircuit. But here we preserve the relative connection probabilities of the Pochance-Diesmann model. This does not stand for Parkinson's disease, but Pochance-Diesmann in this case. So instead of the in degrees that I mentioned before, and the thinking was that you have some areas that have, for instance, vanishing layer 4, a very thin layer 4. But in those areas, they should, if you preserve the in degrees, they should send a large number of synapses to the other population, which might be unrealistic. And it's more realistic, perhaps, that the number of synapses is proportional to the sizes of both the sending and the receiving populations, and that's achieved by preserving the connection probabilities. I say here relative connection probabilities because we're still scaling the total numbers of synapses to achieve the total, to correspond to this volume density of synapses that was experimentally determined. And this model, it's similar in size in terms of number of neurons as the macaque model, but it has more synapses. So human neurons, they're sending and receiving more synapses per neuron. And we use a really old publication to determine the neuron densities. Now we have a collaboration with colleagues in Jülich that are measuring neuron densities and really updating this publication, but the data are becoming available slowly, and they use a different parcellation as well. Here we did distinguish the single neuron parameters of the excitatory and inhibitory cells according to data from the Allen Institute. And again, we investigate multi-scale resting state activity. Of course, for the human brain, we don't use invasive data to determine the connection probabilities, but we use DTI, diffusion tensor imaging data. And these data, they're not layer resolved, so we still need to, we use laminar patterns from the macaque to kind of estimate what they might be like in human. And we again also statistically map the synapses to the target cell bodies using morphological reconstructions of neurons that were here published for human neurons, mostly from the temporal lobe. And it's only the excitatory neurons, so it's again a limited data set. Here are the laminar patterns that, so for the human model, we again have the neuron densities. So we take the relative neuron densities of sending and receiving areas, and that relates presumably to sort of hierarchical ordering and informs their laminar patterns. You see there's a lot of variability unaccounted for that can be investigated in future. Another difference between the macaque and the human models is the fraction of connectivity that goes through the white matter. I mean, we don't explicitly model gray or white matter, but white matter meaning between areas in this case, and gray, let's say, corresponding to within areas. And for the macaque monkey, this has been, there's an estimate available from experiments around 79% gray matter connectivity. But in the human brain, maybe unintuitively, but because we have a larger brain volume, then the relative surface area between the gray and white matter is reduced, and relative to how many neurons need to be connected. and Herculano Hosel derived a scaling law for that, and then we applied this using the macaque data and arrived at that in the human brain there should be about 86% gray matter connectivity. Putting everything together, we can arrive at the population level connectivity matrix, and then we turn on the simulation. And again, when the inter-area synapses have the same strength as the local ones, then the activity does not match the experimental data. But when we increase the strength of the cortical synapses, so the inter-area synapses, then at some point, suddenly, the microscopic spiking activity and the fMRI functional connectivity are well reproduced. So there are not so many spiking data for human cortex, but some were available here from medial frontal cortex, and then we compare the data from the corresponding area in our simulation. And best fit version, the firing rate distribution was nicely reproduced, and also the distribution of two measures of irregularity of spiking. So coefficient of variation of the interspike intervals and this revised local variation that is a measure that kind of controls a bit for differences in spike rates. Just as some illustration of how one might investigate the activity mechanisms in this model, we compared to simulations with identical random seeds, but just introduced one extra spike in the pericalcarine area, V1, more or less, and saw when there is at least one additional spike or one spike missing in each of the other areas and populations of the model. And this is the version of the model where the local synapses have the same strength as the interarea synapses, and this is this best fit version with a stronger interarea synapses. And in both cases, there's a fast spread of these changes in activity across the entire network. So basically, this is some signature of chaos in the network. And how fast this spreads is quite well predicted by the mean delay of the shortest paths on the population level between the different populations. And it was slightly faster in this best fit version. So that brings me to the end already. So in conclusion, these types of models, they can reproduce microscopic and macroscopic aspects of cortical dynamics. The network construction is a useful exercise to reveal gaps and regularities in the data. And the models, they predict stronger interarea compared to local synaptic strengths. And that cortex may use joint clustering of excitatory and inhibitory cells to support robust balance and activity propagation. And we hope that incorporating many regions and features into unified models will contribute to consolidating the model zoo in computational neuroscience. And at least we try to make all the codes openly available and in a hopefully reasonably documented way so that people can reuse it and use it for new studies. That I would like to thank my collaborators and current and former group members, especially the ones highlighted in red. And thanks for your attention, and thanks to the organizers. It was really nice.